So we will get started for today. We're going to continue on with buffers, picking up where we left off on Wednesday. So we were talking about acids and bases in general, and then we did these simple calculations to determine pH of solutions using the M1V1 equation. And the next thing we're going to look at is the idea of buffers. Now, buffers are important in doing experimental biochemistry, as we are dealing with proteins, dealing with a bunch of biomolecules. And these biomolecules only work at certain pHs. And it depends where in the body you're found, what pH you work at, but it's all different. Inside the cell has their own buffers. Our blood has a buffer. So to really understand biochemistry completely, we need to know how a buffer works. And what a buffer is, it's a solution that contains both acid and its conjugate base. If you remember, the only difference between an acid and its conjugate base is one proton. And what buffers do is that they resist pH change. They can react with an acid or a base and not change the pH of the solution all that much. This really only works for weak acids, though. Strong acids can't be part of a buffer. It's only weak acids and weak bases. Buffers themselves, they have an effective range of +1 to -1 of what is called their pKa. The pKa is pH when base concentration equals acid concentration. So if you want to make a buffered solution that has the maximum range of pH it can buffer, what you want to do is you want to ensure that the buffer has equal parts base and equal parts conjugate acid. Then you get +1, -1 pH. So for example, NH4 on the chart roughly has a pKa of like 9.2. So its buffer range is roughly 10.2 to 8.2. Now we can determine the pH of any buffered solution using what's called the Henderson-Hasselbalch equation. Henderson-Hasselbalch says for a buffered solution, the pH of that solution is equal to the pKa of your buffer plus log of base concentration divided by acid concentration. And we're going to work on some examples today using the Henderson-Hasselbalch equation to determine pH of buffered solutions. Now, I will say that the amount of base and acid, if that's the same, that's the maximum range you can get. But in practice, a lot of the times you won't make it 50-50. For an example, if you're doing an experiment and you know there's going to be a lot of acid in your experiment, then you might want to make your buffer with more base in it. Buffer max for +1, -1. Well, that's the maximum range, right? +1, -1 of its pKa. So that NH4, right? NH4 has a pKa of roughly 9.2. So the maximum range it can work as a buffer is either between 8.2 and 10.2. If you go below 8.2, you can see this giant drop. pH drops very fast because the buffer no longer works. If you keep adding acid, it's going to interact with the water instead of the buffer. Conversely, if you go above 10.2 and you keep adding base, that base no longer interacts with the buffer. It'll start interacting with the water and make the pH just skyrocket up. So let's do a question where we look at the Henderson-Hasselbalch equation. And we're going to do this one together. And then question 4, I'm going to give you some time to work on that so you can become more familiar with the Henderson-Hasselbalch equation. So I want to know what the pH of a 1 liter solution is if it contains 10 milliliters of 2 molar acetic acid plus 5 grams of sodium acetate, and their sodium acetate's molar mass. The pKa of acetic acid is 4.76. All right, so when doing word problems, I always like to write out the equation or equations I'm using and declare my variables. It helps me to have a roadmap. So it's always base over acid. I will say, if you look up the Henderson-Hasselbalch in different books or online, you might get this formula, pH equals pKa minus log acid over base. It's the same equation. You'll get the same answer. I use the plus form. That's the one the book uses. So that's the one we're going to work with. But just in case you look it up some other source, they are actually the same equation. It's just math, trick of math to flip the log like that. All right, so let's declare our variables. pH, I don't know. That is what I'm trying to figure out. pKa, given 4.76. Plus log base. I don't have that concentration. Remember, bracket equals moles divided by liters. So my base is sodium acetate. So the concentration of sodium acetate divided by acid, acetic acid. I'm just going to call that ACE. I don't have that concentration as well. It's not 2 molar. So what we're doing here is the first thing we need to do is a dilution. This is exactly like what we did on Wednesday. I'm taking 10 milliliters of this 2 molar acetic acid, and I'm diluting it to a final volume of 1 liter. Right? So let's figure out, the first thing we want to do is figure out what is the concentration of acetic acid in our solution. We can figure that out like we did on Wednesday. M1V1 equals M2V2. I'm going to say the 1 liter solution is M2, or sorry, that would be V2. You could call it V1 if you want, doesn't matter. So 10 milliliters is V1, 2 molar is M1. So I'm taking 2 molar of a 10 meter solution and I'm diluting it. So I don't know the molarity, that's my unknown of my new solution, in 1 liter, which is 1,000 milliliters. So I converted this 1 liter to 1,000 milliliters to do this equation. Solve for M2. So M2 equals 0.02 molar. My concentration of acid is 0.02 molar. All right, before I continue on and solve the base, is there any questions so far about how I set this up, what I did, what my logic is? Did you get lost through any of that? Any questions at all? Right. If not, we can move on to finding base concentration. Right. So here I have a weight of base, but I need a concentration, which is moles divided by liters. So I have to figure out how many moles of sodium acetate I have. Hopefully we remember that skill from general chemistry. If you have a weight of a molecule in its molecular weight, we can do easy conversion where I'm going to take 5 grams divided by 82 grams to get moles. So this is my train track method for doing unit conversions. If you have a unit at the top and you have the same unit at the bottom, those units cancel out. I'm left in units of moles. Multiply everything on the top together, divided by what's on the bottom. So this will be 5 divided by 82. And in my solution, I have 0.0610 moles of sodium acetate. So this is my base. However, I need molarity. I need concentration, not moles. So I need to take this moles and divide it by my volume, which is simple here because my volume is 1 liter. So the number of moles I have is actually the same as my molarity. That's not always going to be the case. It just happens when I made this problem, I did that. Now we can go and we can solve our equation. pH equals pKa, which is 4.76, plus log of our concentration of base, 0.0610, divided by my concentration of acid, 0.02. My pH equals 5.24. That is the Henderson-Hasselbalch equation in a nutshell. All right, now that you've seen all that worked out, does anyone have any questions? Anybody want me to go over something that they might have missed? Got stuck on something? Not sure how I got a number? Let me know. All right, so one thing I do want to point out, with doing math, especially everybody, this is a level 4,000 level class. So you are, you're advanced students. You've been in college for a while. If not, some of you have probably graduated college and are coming back just for biochem. When doing math problems, when getting answer to these types of problems, you should always ask yourself, did I expect that answer? Does my answer make sense? It is not sufficient to just trust your calculator and whatever your calculator says. You're like, oh, okay, this is what my calculator says, so it must be the right answer. Everybody here is smart enough to start adding logic to what their answers are. By that, I mean, if we look at our concentrations, our concentration of base is greater than our concentration of acid. So in our buffer system, we have more base than acid. So if we look at our pH, our calculated pH should be greater than our pKa. Remember, pKa, that is the pH when concentration of base equals concentration of acid. All right, that's what pKa, one of the things pKa means. So if you have more base than acid, when you use Henderson-Hasselbalch, your pH should be greater than your pKa. Conversely, if you have more acid than base, your pH should be lower than your pKa. So that kind of next level thinking we need to train ourselves, not as students. Train your ability to think like that, because the greatest thing you can learn getting out of college is learning how to think and learning how to study, and just asking yourself, do these numbers make sense? When I'm getting out, does that logically make sense? It's a very important skill and a very hard one to train. Not a lot of people have that when it comes to math. So be on the lookout and see if you can train yourself with that. All right, so if there's no questions about this particular problem, I will give you some minutes to try and struggle through this one. So if the pH of acetic acid sodium acetate solution is 4.25, what's my ratio of base to acid? So everybody take a few minutes to tackle this. If you have questions or if you get an answer and you want to check with me, let me know through a chat. You can always send me a private chat, a private message. Otherwise, I'll be back with the answer in a couple minutes. However, please do try and work on this, as working through problems, struggling is really how you learn. All right, so hopefully you had some time to at least attempt this or think about it. Let's take a look at this. So rewrite my equation and start filling stuff in. pH equals pKa plus log base over acid. So my pH is 4.25. My pKa is 4.76 plus log base over acid. So subtract pKa from both sides, which is negative 0.51 equals log base over acid. Now, when you have a log of something, the way to get rid of a log is you do 10 to the power of each side. And I'm just going to double check my calculation before I continue, because I have a bad habit of miscalculating stuff. Okay. So I take each side 10 to the power. 10 to the power of log cancels out. 10 to the power of negative 0.51 is 0.309. So my ratio of base to acid is 0.309, which again makes sense, right? My pH is lower than my pKa. Therefore, I should have more acid than base. Therefore, if I'm taking base divided by acid, or ratio of base to acid, I should expect a number lower than 1. If you didn't get a number lower than 1, that should be a sign that you messed up somewhere, miscalculation somewhere. All right, so with that, any questions about question 4 here? All right, so I believe that's the last slide of this PowerPoint. So we'll go on the PowerPoint for the 29th, which is today. Happy Friday, everybody. Oops, forgot to share it. All right. So we are going to start and learn about nucleotides now. So we're finally getting into our first biomolecules. DNA is the keeper of information. DNA is what directs a cell to make proteins and everything. So it makes sense on our journey through biochemistry, the first biomolecule we're going to look at is DNA. So what I'm going to ask you to do for our nucleotides is learn how to recognize the bases. For the tests, because the tests are all online, I'm not going to ask you to draw anything, but I am going to ask you to recognize things. So you need to be able to tell the difference between A, G, C, U, and T just by looking at them. I guarantee you 100%. I'm going to give you a question where I show you like a base and ask what is this? You also need to know, how do bases connect to the sugar? So if we look here in the middle, this is one of the sugars that can be used for nucleotides. This is called a ribonucleotide, which is part of RNA. I know the words are cut out, but this is a deoxynucleotide on B, which is DNA. Deoxy, because if you look at the 2' position, there's no oxygen, so deoxy. RNA does have the OH. Here's the ribose sugar, and here's the deoxyribose. So the bottom right pictures are showing you what the sugars look like if there's no base attached. the A and B pictures are showing you what it looks like when the base attaches. So the only difference is that your base attaches to the 1' sugar carbon. So that's where your base goes. And the way it connects, if you look on the table, you see these little X's. These X's are showing you this is the atom that connects to our sugar. So you form what's called a glycosidic. This is called a glycosidic bond. You form a glycosidic bond between your sugar and your base. And the last thing we need to know is what's a 5' nucleotide and a 3' nucleotide. That's shown A and B as well. When we're talking about strands of nucleotides, the top of the strand and the bottom of the strand are different. The top of the strand has a phosphate on the 5' carbon. So that's why it's called the 5' end. The 3' carbon has a phosphate on the 3'. The phosphate on the 3' carbon. So the 3' sugar. This directionality matters a lot. Enzymes work on going certain directions, so they can tell the directions. So that's what we need to know for our test one. So another good old memorization. Not too bad though. There's only 5 bases. And we also have to be able to name our nucleotides based on the phosphates we have. So here in my example, I have ATP. So it has adenine as our base. We have ribose as our sugar. You can tell it's ribose because of this OH. And then we have 3 phosphates, 1, 2, 3. So see if, and I'll give people like a 30 seconds or so to determine what are the 2 things I'm showing there on the bottom then. What are those called? And this is another example of a question you might get. So for A, GDP. For B, well, it's the same base, cyclic AMP. Monophosphate, it's only 1 phosphate. I know it's cut off for you a little bit there, but it's cycling around. So that's CMP. If you remember from your general biology, ATP is the energy currency of the cell. It's what the cell mainly uses for energy. CMP is what happens after you use both of the phosphate for ATP. CMP, cyclic AMP, is a signal that the cell is low energy. CMP will go around to different proteins in the cell and turn on, like glycolysis, turn on the Krebs cycle. So while we're looking at these nucleotides, I do have a critical thinking question, another one of these critical thinking questions. Inside your body, inside your cell, you have ATP, you have TTP, you have CTP, you have GTP, and you have UTP. Can anyone come up with a hypothesis as to why we use ATP for energy compared to everything else? there is a caveat there. We do use GTP in some enzymes, but the majority of enzymes use ATP. Can anyone come up with a hypothesis of why we use ATP, not like CTP, UTP, TTP, or GTP more? Anyone brave enough to share thoughts? And you can always share it privately to me. I won't name you. Adenine is most abundant. That's a good hypothesis. I think naturally they should all be the same because we make them. So in just talking about evolutionary terms, I don't see a reason why way back then adenine would be more abundant than the other ones naturally, but I can't disprove that either. So that's a great hypothesis. Glycolytic pathway. How so? What do you mean by glycolytic pathway? The bonds in ATP are unstable since they contain more energy. I would say that the energy of ATP is the same as GTP, CTP, UTP, and TTP. Good idea. I like how you're thinking about energy in terms of bonds, but they should be relatively all the same because the energy comes from the 3 phosphates connected. Anyone else? I'll give people like maybe 30, 45 seconds if you want to churn your brains. Make your brains hurt by thinking. In the glycolytic pathway, ATP is synthesized by substrate level phosphorylation. Okay, why isn't it GTP or CTP or TTP? I guess that would be my question. Why is that important? Why didn't glycolysis pick something else? That is a true statement. That is what happens in glycolysis. But why? So my idea, and I haven't really researched this, so if anyone wants to research and prove me wrong, I'll definitely give you brownie points, extra credit if you want to prove me wrong about this. It's more or less luck of the draw, as in nobody knows, as far as I know. That's a very hard question to answer, and it's questions I will ask throughout this semester where I'll pose questions to you guys that don't have an answer because I just want to see how you're thinking. Does it have anything to do with ATP being able to bind U and T? Nope. ATP does not interact with UTP or TTP, really. At least I don't think so. I mean, that could actually, now that you bring that up, that could be possible as an explanation, in that people think all biomolecules started with RNA. RNA has U, So maybe that's why there was an A, but you could, now that I say that out loud, you can make the same argument about any nucleotide. But again, good idea. I like how you're thinking about that. Yeah, so my hypothesis is it's just kind of random chance in that it makes sense to use only one triphosphate as energy, right? If I'm an organism and I have 1,000 different proteins, and these proteins need energy, it is much easier if they all use the same energy source, right? If they all use ATP, then I can regulate them easily. They can respond to levels of ATP versus CAMP. So to me, that makes a lot of sense. Granted, we do have some proteins that use GTP as energy because they are regulated differently. Why ATP instead of CTP? I have no idea. And going through this class, I'm sure it happens every semester. Somebody will ask me questions that I have no idea to. And that's one of the beauties of science, is that we still don't know a lot. And it's an exciting time to be a scientist. I think I read somewhere that like 90% of all scientists who have ever lived are alive right now. This is a scientific revolution, and you all are getting in at the ground floor. So if anyone wants to do research as to why ATP is picked over everything else, send it to me. I'll be happy to give you like some brownie points. But that was just a little diversion, a little thing to keep you motivated during the dark days of your studies. But on the test, you need to be able to look at the different nucleotides and tell me what's what. Yep. A lot of things in science make you go, why does this work? And then you look into it like, oh, nobody knows. All right. So let's look at how nucleotides are actually put together now, right? So we use ATP, single nucleotides like that. Most of the time, they're going to be in bonds, though, in nucleic acids. The way that nucleic acids are connected together are by their 5' and 3' carbon. They're connected through this phosphate. So every time you have a nucleotide, you have a phosphate that connects the 3' and 5' carbons. Phosphates are negatively charged, so that's why DNA is incredibly negatively charged. Excuse me, the whole backbone is negative. Scientists, we have a system where anytime you see a DNA strand presented, no matter what form it's in, is it drawn out, is it in letters, it is always 5' to 3'. And the 5' end is where you have one phosphate, 3' end. You'll usually have an OH there. It's possible to have a phosphate, but that's very rare. It usually ends in an OH. And most of your enzymes that read DNA will read it 5' to 3'. Not all of them. There are examples that go reverse, but a lot of them go 5' to 3'. When we connect amino acids, when we connect anything, they're called, the polymer that's made is a oligomer. So that's how you say that word, oligomer. RNA, typically single-stranded, not always. There's double-stranded DNA, RNA out there. DNA, usually double-stranded, not always. There's a lot of examples of single-stranded DNA. And in most organisms, we follow Chargaff's rule, where the amount of A in your cells equals the amount of T, the amount of G equals the amount of C. That's because of hydrogen bonding. A bonds with T, G with C. There are some organisms that don't follow this rule. They're usually in weird environments or viruses. Weird environments, I mean like high heat, high acid. But the majority of organisms do follow Chargaff's rule. So one of the things that I would like you to be able to do, and unfortunately, I can't test you on it this semester by having you draw. I can only test on by having you recognize. So one of the perks of being an online class, you don't have to actually do this question on the exam. But one thing you should be able to do from what we just talked about is have this table in front of you and draw a Cu RNA dinucleotide. So just to get a little bit practice in of how everything's connected again, try that. I'll give you a few minutes and piece of paper. And unfortunately, I can't see your piece of paper. Try and draw me what a Cu RNA nucleotide looks like based on all the connection stuff we just talked about. So see if you can remember how everything is connected. So I'm just going to start slowly filling stuff in here. So it's RNA. So make sure you draw RNA, not DNA. Remember, RNA has the hydroxyl on the 2' position. Numbering goes 1', 2', 3', 4', and there's a carbon out here. That's 5'. So I need 2 RNAs. This is my 5' end. So my 5' end gets a phosphate. They are connected by a phosphate. The 3' end just has a hydroxyl. Then my bases, first we have C. So we have an N there. I'm kind of bad at drawing these. Oh, running out of space, NH2. And there, double bond O. And then a U. Double bond O. So that's a Cu RNA dinucleotide. RNA, usually not double-stranded. And usually, again, I'm not going to ask you to draw that. I was going to say usually you only need to draw 1 strand for me, but you don't need to draw it all this semester. So this is a good way to just test yourself if you understand how everything's connected together. Even though this is close, like that's just me bad drawing, 2' not connected to base below it. It's just, I didn't space out my molecule too well. All right. So talking about double helixes, because our nucleotides are normally in a double helix, you have 2 nucleotide chains that wrap around each other. And they're anti-parallel. One runs 5' to 3' in one direction. So like this one will go 5' to 3' down this direction. The other one is the opposite. 5' to 3' in the other direction. That's what anti-parallel means. Because of the way they wrap around each other, the DNA bases are exposed at different widths that repeat. So you have one area called the major groove, where the DNAs open up a little bit more. The minor groove is as these bases aren't as exposed. So it goes major groove, minor groove, major groove, minor groove. The bases themselves, they are pointed towards the center of the helix, where they are hydrogen bonding. The sugar phosphates make up the backbone. They're on the outside. This model, not obvious. I mean, now it's obvious that we've seen it, but like discovering it took a couple decades, not easy to come up with. The beauty of this model is that as long as you have one strand, you always know what the other strand should be, which is perfect when cells are dividing. You can just give each new cell one strand of DNA. You have an automatic template. That's because A goes with T, C goes with G. You can have different types of combinations. Here I have single-stranded RNA. And what single-stranded RNA will do is that it will bind itself. And RNA can create some really interesting, unique conformations. DNA, boring, double helix. There's like 3 different types of double helixes it can form, but it usually only forms double helixes. RNA, unlimited possibilities. Because it binds within its own single strand, the number of different shapes you can get is like equal to the number of different proteins you can make. It's just such a big number, you can't even begin to understand like just the number of possibilities through 4 different bases. So my last question of the weekend is thinking about the biophysical properties of DNA. So biophysics is what I studied. So a lot of the questions I ask in biochemistry I can't speak anymore, are a physical nature. Okay, so I'm curious now too. I just reviewed some notes I had. ATP hydrolysis drives many unfavorable reactions to completion. So that's why ATP acts as the energy currency. ATP hydrolysis provides the energy needed for glucose phosphorylation. So really, I think it's favored because it relates to a lot of energy. GTP and such don't have what it's take. I'm pretty sure GTP has the same energy as ATP. The energy is in the phosphate bonds. It has nothing to do with A. CTP should have the same energy as ATP as well. The enzymes themselves have been specialized to only interact with the adenosine now, right? Because evolution, if we have a lot of ATP around, of course we're going to use ATP. But if you're just talking about thermodynamics, I can't see any reason why ATP is much more favorable than CTP. They should have the same energy. They have more energy than GDP, yes, but not GTP. All right. So getting back to our question then. So we have DNA. Why is it easy to separate DNA at pH greater than 11? Why, if I make my solution basic, my DNA falls apart? Does anyone know the answer to that? Why, when I increase pH, and this is a common method for separating DNA, you just increase how basic your solution is. It breaks hydrogen bonds. Exactly. If you don't know the answer to something in biochemistry, it's probably hydrogen bonds, right? So hydrogen bonds are what keep DNA, double helix DNA together. As your solution becomes more basic, the bases themselves loses hydrogens and they can't hydrogen bond anymore. So it's breaking the intermolecular hydrogen bonding, intermolecular H bonding. Okay, good. B is a little more tricky. Why, when I increase the salt concentration, is it harder to separate DNA? That is why when I add salt, do I need to put in more temperature, more heat to separate DNA? B is a thinker. Does anyone have any hypotheses for B? Why there's a correlation? The higher the salt concentration, the higher the temperature needed to separate DNA. What is the hypothesis for that one? Our last hypothesis of the week. NaCl is a good energy conductor. NaCl is a great conductor of electricity. Fun fact, water itself does not conduct electricity. It only conducts electricity with ions dissolved in it. So NaCl can conduct electricity. That's true. Very true. What's that have to do with DNA separating? Or does it have something to do with DNA separating? Here's a hint. What is the overall charge of DNA? Is DNA negative, positive, or neutral? It's negative. It's very negative, right? All right. Here's DNA strand number 1. Here's DNA strand number 2. They're both negative. What happens when you put one negative molecule next to another negative molecule? Do they want to come together or do they want to pull apart? Repulsion, they want to pull apart, right? So DNA has 2 different forces acting on it at all times. They have repulsion. Since the DNA are negatively charged, they don't want to come together. I know that's weird to think about because we think of double-stranded DNA. DNA strands don't want to come together, except they have another force acting on it. Hydrogen bonds in between the bases holds them together. So when you're thinking, will my DNA be double-stranded or single-stranded, you are asking yourself, which force is more important? Which force is stronger? Hydrogen bond or repulsion? As you increase temperature, what you're doing is that you're increasing atom movement. These bases are moving much faster. Like they're vibrating a lot faster as you get hotter. And so their hydrogen bonds become weaker. So at higher temperatures, H bonds become weaker. So that's why DNA melts at high temperature. The thing with salt is made out of 2 things. Depending on what salt, let's just use sodium chloride as an example. Positive sodium, negative chlorine. Positive and negative attract each other. So if you have a lot of salt in your DNA, all of these salt molecules are going to go, all these positive molecules are going to go and interact with the negative phosphates. What that does is that it greatly lowers this repulsion force, because in essence, you are decreasing the negative charge of the strands. Decreasing is not the right word. You are screening the negative charges. It's called electrostatic screening. basically, if you have a lot of positive ions around your negative ions, and the negative ions don't know they exist anymore. That is, the negative ions on each strand can't see each other anymore. They don't feel each other because they're being more or less neutralized. Therefore, since you don't have repulsion anymore, it takes more energy to break those hydrogen bonds. Energy is heat. It takes more heat to break the hydrogen bonds. That is why, on a biophysical level, if you add more salt to your DNA solution, you need to increase the temperature to melt it. It's because you're hiding the negative charges from each other. And so you don't have repulsion naturally trying to break your DNA, and you need to have more heat to break the hydrogen bonds than you would normally. That makes sense? Kind of, a little bit, somewhat. Well, with that, it is 2:56 on my clock. So I will end it there. If you have questions about anything that we went over, always feel free to email me. I will put homework up under Wiley Plus like normal. Other than that, hope you have a good weekend, and I hope to see you all next week. All right. Take care, everybody.